Hi everyone, my name is Noah Burkett and I'm a senior biology and Spanish double major. For this presentation, I'll be outlining my research with Dr. Patuk from last summer. We studied the effect of the mutation from asparagine, abbreviated N, to aspartic acid, abbreviated D, at residue 185 in the N-terminal domain of MPC1, or Niemann-Pick type C1 protein. NPC1 is a large multi-domain protein essential for transporting cholesterol from late endosomes and lysosomes to the endoplasmic reticulum and other cellular compartments. NPC1 is known to form a complex with NPC2 to bind cholesterol from the membrane and then transport it inside the membrane. Mutations in NPC1 cause NPC disease, a lysosomal storage disease characterized by intracellular accumulation of cholesterol. The mutation mentioned from asparagine to aspartic acid at residue 185 has not been proven to be deleterious or disease-causing, although it's thought to be due to its conservation among orthologous sequences. The prevalence of this mutation is within the range that suggests that it isn't so common that it's a benign mutation, but common enough to study its effects. The main hypothesis of this study is that the mutation from asparagine to aspartic acid at residue 185 will be deleterious based on changes in specific interactions, RMSF, and binding and folding energies. We used 3 states to test this hypothesis: the apostate with just MPC1, a complex with MPC1 and cholesterol, and lastly a complex with MPC1, MPC2, and cholesterol. After the effects of mutation are studied, virtual screening will be used to test FDA-approved drugs and other small molecules to determine if any drug therapy that exists can be implemented for this mutation. Figure 1 at the bottom left of the poster shows the complex with MPC1 in red, MPC2 in blue, and cholesterol in green. The mutation site is highlighted in yellow and is highlighted in yellow throughout all the rest of the figures as well. Table 1 shows the changing and binding and folding energies due to the mutation. With a decrease in folding energy, outlined by 2 other predictors, the protein will be in a less stable conformation. The increased binding affinity, predicted by the last tool in the table, will affect the interacting of NPC1 and NPC2, making it more difficult for the complex to separate. Next, in Figure 2, we looked at RMSF analysis of the apostate, MPC1 with cholesterol, and the complex of MPC1, MPC2, and cholesterol, respectively from left to right. RMSF, or root mean square fluctuation, shows the changes in areas of the protein due to mutation. In the apostate and in the NPC1 cholesterol duo, we see an increase in flexibility in most of the protein. The darker the color, the more significant change of flexibility. The complex showed an increase in rigidity in NPC1, where it interacts with NPC2. Next, in Figure 3, we looked at hydrogen bonds, a type of specific interactions. Overall, in all 3 states, there were an increase in hydrogen bonds. However, decreases in hydrogen bonds in certain loops and regions will cause less rigidity in those areas while the rigidity of the entire system will increase. Figure 4 shows salt bridge analysis, another type of specific interaction. There were increases in the number of salt bridges when NPC1 is not in the apostate, showing an increase in rigidity. However, there are still areas of net increase and net decrease of salt bridges, showing the respective increase in rigidity and flexibility in the mutant type. Virtual screening analysis showed potential small molecules such as FDA-approved drugs and small molecules that could be used as a potential treatment to restore wild-type properties in the mutant type. Currently, Dr. Swain's research lab is testing some of these drugs in a yeast paralog of NPC1. Overall conclusions from my study are that the mutation at residue 185 from asparagine to aspartic acid can be deleterious due to the specific changes in specific interactions, RMSF, binding energy, and folding energy, in conjunction with the conservation of the residue, and that they are likely candidates for drug therapy to restore wild-type properties to the mutated protein. Further testing from molecular dynamics simulations and in vitro trials will support this. Thank you for listening to my presentation, and I would like to thank Dr. Patuk for advising me, S.E. Embry and Dr. Gordon, S.E. Embry for funding my research, and Dr. Gordon for leading that program, as well as Dr. Rishbeater for leading the Summer Fellows program, which allowed us to live here over the summer. Thank you so much. Have a good one.